Welcome. Today, we're diving into the fascinating world of gene regulation in the budding yeast, Saccharomyces cerevisiae. This tiny organism has played a huge role in helping scientists understand how genes are turned on and off. Recent research has revealed that transcription initiation, the process where gene expression begins, is far more dynamic and widespread in yeast than previously thought. By mapping transcription start sites at single nucleotide resolution, scientists have uncovered a complex landscape of gene regulation that responds to environmental changes. Let's explore how these discoveries are reshaping our understanding of gene expression. Saccharomyces cerevisiae, commonly known as budding yeast, is a classic model organism. For decades, it has been used to uncover fundamental principles of genetics and cell biology. Its relatively simple genome and ease of manipulation make it ideal for studying complex processes like transcription initiation. By focusing on yeast, researchers can systematically investigate how genes respond to different environmental conditions, providing insights that are often applicable to higher organisms, including humans. A major breakthrough in this study was the use of NNTICAGE sequencing, a technique that allows researchers to map transcription start sites, TSSs, at single nucleotide resolution. By growing yeast in 9 different conditions and sequencing millions of RNA tags, scientists identified nearly 1 million well-supported TSSs. This comprehensive mapping revealed that transcription initiation is highly pervasive, occurring throughout the compact yeast genome, and not just at a few select sites. At the heart of transcription initiation are core promoters, specific DNA regions where RNA polymerase II binds to start gene expression. This study identified core promoters for about 96% of VURAFUD protein-coding genes in yeast. By integrating TSS data from multiple conditions, researchers could correct previous missensations and even suggest alternative start codons for some genes. This level of detail is crucial for understanding how genes are regulated at the most fundamental level. One exciting outcome of this research was the correction of translation start codons for 122 genes and the suggestion of alternative start codons for 57 more. By comparing transcription data with protein evidence and evolutionary conservation, scientists improved the accuracy of the yeast genome annotation. This not only helps in understanding yeast biology, but also sets a standard for genome annotation in other organisms. Study found that transcription initiation in yeast is much more widespread than previously recognized. Over 4 million unique TSSs were identified, with about 1 million supported by multiple sequencing tags. Interestingly, most transcription starts in non-coding regions, and only a small portion of TSSs are active in all conditions. This highlights the dynamic nature of gene regulation in response to environmental cues. Yeast cells constantly adapt to their environment by changing which genes are active. The study showed that 56% of yeast genes are controlled by multiple core promoters, and the usage of these promoters shifts depending on environmental conditions. This alternative promoter usage allows yeast to fine-tune gene expression, ensuring survival and optimal function under stress or changing nutrient availability. Striking example of promoter shift is seen in the CIK1 gene. Under normal conditions, CIK1 transcription starts from a distal promoter. However, when exposed to mating pheromone, transcription switches to a proximal promoter, producing a shorter protein isoform. This shift not only changes gene expression levels, but also affects protein function, demonstrating the biological significance of dynamic promoter usage. Researchers classified yeast core promoters into 2 main types, constitutive and inducible. Constitutive promoters are always active, regardless of environmental conditions, while inducible promoters are only active under specific circumstances. About 55% of promoters are inducible, showing strong condition specificity, while 45% are constitutive, ensuring essential genes are always expressed. Activity of core promoters is closely linked to chromatin structure. Constitutive promoters are typically found in nucleosome-free regions, making them more accessible for transcription. In contrast, inducible promoters are often wrapped in nucleosomes and require chromatin remodeling to become active. This difference in chromatin landscape helps explain why some promoters are always on, while others are tightly regulated. Promoters can vary in shape, from sharp, where transcription starts at a precise point, to broad, where initiation occurs over a wider region. The study introduced the promoter shape score, PSS, to quantify this. Constitutive promoters tend to have broader shapes, supporting consistent gene expression, while inducible promoters are often sharp, reflecting their specific activation under certain conditions. Both constitutive and inducible promoters show a strong preference for pyrimidine, purine dinucleotides at the transcription start site, especially CA, TA, and TG. However, constitutive promoters have an even stronger preference for these motifs, which may contribute to their higher transcriptional activity. Unique sequence features, such as the presence of TATA boxes or GA elements, further distinguish promoter classes. Shifting core promoters doesn't just change where transcription starts, it can also alter the length and structure of the resulting mRNA's 5' untranslated region, UTR. These changes can impact mRNA stability and translation efficiency, ultimately affecting how much protein is produced. This adds another layer of regulation, allowing yeast to adapt quickly to environmental changes. Discoveries made in yeast have broad implications for understanding gene regulation in all eukaryotes. The prevalence of alternative promoter usage and dynamic transcription initiation suggests that similar mechanisms may operate in more complex organisms. Future research will likely focus on identifying novel genes, understanding the functional roles of low-activity promoters, and exploring how promoter shifts influence cellular adaptation. Summary: transcription initiation in yeast is far more pervasive and dynamic than previously recognized. By leveraging advanced sequencing technologies and analyzing multiple growth conditions, researchers have mapped a complex regulatory landscape that enables yeast to thrive in changing environments. These findings not only enhance our understanding of yeast biology, but also provide a blueprint for studying gene regulation in other organisms. Journey into the dynamic world of transcription initiation continues, promising even more exciting discoveries ahead.